Okay, let's get started. Let's move on to our first company for today, which is Alterity Therapeutics, trading into the ASX code ATH with a market capitalization of around $16 million. Alterity is a clinical stage biotechnology company dedicated to creating an alternate future for people living with neurodegenerative diseases. Presenting for today for the company, we have Dr. David Stamler, the CEO. David, please feel free to take it away.
Well, thank you, Peter, and thanks to everyone. Good afternoon, and appreciate you attending our discussion today. As Peter mentioned, I'm the CEO of Alterity Therapeutics, and I'm really excited to tell you about the progress that we've made in the exciting 6 months that are ahead of us. Next slide. These are our forward-looking statements, which we encourage you to review. Next. So Alterity Therapeutics is a clinical stage biopharmaceutical company, and we are focused on developing disease-modifying treatments for neurodegenerative diseases, such as Parkinson's disease and related disorders. We've recently disclosed positive data from an open label study that confirms our clinical development approach, and I'll be reviewing those data with you on the call today. Our flagship study is our phase 2 double-blind trial that is ongoing and expected to read out in about 5 months. So something very exciting to look forward to. For our lead indication, which is a disease called multiple system atrophy, we have orphan drug designation in the US and EU, which gives us various benefits, including tax benefits and exclusivity. We have a strong patent portfolio from our ongoing discovery efforts, and we have significant R&D experience within the company, and in particular, I have 3 FDA approvals under my belt in the area of neurology. Next slide. So I'll be talking about Parkinsonian disorders today, and just a bit of background. Parkinsonism is the syndrome of motor symptoms that we've probably all seen in friends or family members, and that's a slowed movement, stiffness, or tremor. And this is a major source of disability, and it gets its name from Parkinson's disease, which is the most common cause of that motor syndrome. Next. However, there are other neurologic diseases referred to in general as Parkinsonian disorders that also have this motor syndrome. And 2 of these are called multiple system atrophy, or MSA, and another disease called PSP. Now, our lead indication is multiple system atrophy, which is a rare disease without approved therapy. And I think what's interesting, as you'll see in the subsequent slides, that both Parkinson's disease and MSA have similar underlying pathology, and we are targeting that pathology with our drug. Next. What we'll be talking about is the first 2 entries on our slide here. This 201 study is an early stage study in our lead indication. The 202 study is in more advanced patients. Both these trials are ongoing, and it's the 202 study where I have interim data to share with you. We've recently completed a animal study in primates in Parkinson's disease that I'll share with you in a bit. Next slide. So this is a study of the brains from patients with Parkinson's disease or multiple system atrophy. And on the graph, we see the patients are marked in blue with age-matched controls in green. And what we see is in the areas of pathology in both Parkinson's disease on the left and in multiple system atrophy on the right, that there's elevated iron in the areas of pathology. Now, these are, for those of you who are not familiar with neuroscience, these are the areas that are affected in these diseases. So what we're aiming to do is target this excess iron, which is really contributing to the ongoing pathology and neurodegeneration in these individuals. Next slide. So our drug is called ATH 434. I'll likely refer to it sometimes as 434. It is a small molecule drug candidate that decreases pathologic protein aggregation. Now, the protein itself is called alpha-synuclein. What's important is that by targeting this excess iron that I showed you in the previous slide and redistributing it with the drug, we aim to reduce that protein aggregation and preserve neurons. Now, the drug itself, because it doesn't target a specific disease, it targets the iron, has potential to treat various Parkinsonian disorders. And a word of an important note is that we have been discussing our development strategy and our development program over the last several years with the FDA and the European Medicines Agency. Next slide. So our overall approach is summarized on this slide, as I've alluded to. Our goal is to bind and redistribute the excess iron in the central nervous system. We then want to, the net effect of that is to reduce this alpha-synuclein protein aggregation and oxidative stress, both of which contribute to cell death. And the aim there is to rescue brain cells and neuronal function, with the overall result of modifying the underlying cause of the disease. Next slide. So this is the primate study that I mentioned earlier. And it's a bit of a complicated slide. But if you notice, there's baseline. The first set of open dots are baseline. And then after 12 weeks of treatment, after neurologic injury occurred, animals received 12 months of treatment with either 434 in 2 different doses. Light blue is the low dose, dark blue is the high dose, or placebo, which is in black. And what we see, lower scores are better on this scale that looks at various behaviors and functions in these animals. And what we see is that 434 treatment was associated with improved scores in all animals that received the treatment compared to only 2 of 3 on the placebo. What's important also is that those animals that improved the most had the lowest iron levels, thus reinforcing our strategy of targeting iron to preserve neurons and preserve function. Next slide. All right, so now let's turn to multiple system atrophy, our target indication. Next. Before I talk about the science, I just wanted to highlight the commercial opportunity for this therapeutic, which is significant. With relatively modest assumptions, we've estimated peak sales up to $1.1 billion for MSA alone, and that's in the US alone. And this is because the clinicians who we surveyed, 30 US neurologists, recognize the substantial unmet need for treating these patients. They had a strong intent to prescribe because the drug itself not only targets the symptoms, but targets the underlying cause of the disease. And they really appreciated the fact that this is an oral drug that's taken twice a day, whereas many new therapeutics in neurology area have to be given by injection. Next slide. So multiple system atrophy itself, or MSA, is a rare, highly debilitating and rapidly progressive disease. We see a picture of this on the right, where patients do experience, nonspecific symptoms, and then within a couple of years of disease onset, they start demonstrating Parkinsonism. Now, this is a Parkinsonian disorder that has no approved therapy. As mentioned, it is an orphan disease with estimates up to 50,000 patients in the United States. The disease characteristics, I mentioned the Parkinsonism, but these patients also have the uncoordinated movements, which leads to balance problems and falls. They also significantly have autonomic dysfunction, which is the part of the nervous system that helps you maintain your blood pressure and normal bowel and bladder function. And finally, in addition to these various morbidities, the median survival for these individuals is only 7.5 years after symptom onset. So there's really an urgent need to find and develop new therapeutics that address the underlying pathology of the disease. Next slide. So I now want to talk about the clinical trials. As mentioned, at the beginning of my presentation, the 201 study is our randomized double-blind placebo-controlled study. That's in early-stage patients. And that trial completed enrollment last November. It'll complete this November with data in January. We looked at various biomarkers that are summarized here. I'll tell you about some of those in the next study, both MRI biomarkers, fluid biomarkers from either plasma or spinal fluid, and then wearable movement sensors, which can objectively assess the motor function. We also looked at the various clinical measures that are going to be necessary to support the approval of the drug. Now, the study on the right, which is a much smaller study, is an open label study in a more advanced patient population with very similar biomarkers. And that's what I want to show you on the next slide. So just about 3 weeks ago, we disclosed interim clinical data from this trial, which confirms that our development approach is the right one. Now, of the 10 patients enrolled, we had preliminary data on 7 patients. And these patients were treated with 75 milligrams, and we have data through 6 months of treatment. Now, the first endpoint that I want to tell you about is something called the UMSARS, the Unified MSA Rating Scale. And this really looks at the activities of daily living that you see in the box on the right. And these are all the areas that are adversely affected in patients with MSA. And we saw on this scale that 43% of patients improved over 6 months, and that the change in the overall group of patients compares favorably to patients with similar disease severity who received no treatment. So this is the first positive indication that the drug really has potential to benefit these patients. Next. We also looked at global impression of change, which asked patients or their treating physician to rate the overall neurologic symptoms compared to just before starting therapy. And for both scales, 29% of patients reported improvement in their overall neurologic symptoms. So when we take these two scales, the UMSARS scale and the global impressions of change, we define clinical responders as that patients who improved on all of these. That'll become important when we review the biomarkers on the next slide. So these are some of the measures that we looked at. We measured brain iron using specialized MRI techniques, and we showed that iron content in the substantia nigra, if you recall, that was an area that's affected both in Parkinson's disease and MSA. It was stable over 12 months in those clinical responders. We also looked at brain volume in the areas affected in MSA, and we showed that brain volume was stable between 6 and 12 months in those clinical responders. We also looked at a metabolic marker called myoinositol, which is a marker of the pathology of the disease. The glial cells are the support cells that help nourish and provide normal support for neurons. Those are significantly affected in this disease. And we showed that this myoinositol, which is a marker of pathology of those cells, actually did better than in the clinical responders compared to those that deteriorated. And we also looked at a protein called NFL, which is a marker of neuronal injury. And we showed that on average, those levels were stable in the clinical responders, whereas in the patients who deteriorated, those levels increased significantly. Next slide. So to just summarize the results from this interim analysis, 43% of patients had reduced disability on activities of daily living. 29% had stable or improved overall neurologic symptoms. Objective biomarkers, importantly, demonstrated improvement that was consistent with the clinical findings, and that in general, ATH 434 was well tolerated and had no drug-related serious adverse events. Now, one important note from this study is that the clinical response that we observed, or clinical response was observed in this disease, which is normally a rapidly progressive and unremitting disease. So that was one important finding. The other is that the patients who did stabilize or improve were early stage patients. And that's really important because the next trial that we'll read out in January is really targeting those early stage patients. Next slide. So these are our key milestones. I've told you a little bit about the preliminary data from the 202 study that read out in July. We'll be making scientific presentations in September through November of this year. The 201 study, the double-blind trial, will complete in November, and then we will be releasing top-line data from that study in January of next year. Next slide. So to summarize, we're targeting an orphan disease called MSA that has no approved treatment. We have positive interim data from our open label study, which confirms our development approach. We're expecting top-line data in January of 25. And we have a development team that has multiple FDA approvals. And I thank you for your attention and look forward to any questions you may have.
David, thank you so much for your time. We do have a few questions that have come through. The first is, can you paint some color on the existing partnerships that you have? And do you have any potential partners in the near future?
Yeah, so we're always talking to pharma companies, big and small, that are interested in neurologic assets. I think many of your listeners may know that pharma has largely outsourced discovery and development of early stage projects to small companies. So we have had longstanding relationships and we get frequent inbound inquiries. So that's definitely something that we are continuing to evaluate as we progress with our clinical trials.
Perfect. So just looking at your balance sheet, your cash balance is sitting at around 12.6 million. Would this amount be sufficient to cover expenses in the mid to future, or will you look to seek funding elsewhere?
Yeah, so thank you for that question. Yeah, between our cash balance and our projected R&D rebate for this coming fiscal year, we are funded to complete our ongoing clinical trials.
Perfect. Okay, so the last one is, what are the next steps for Alterity Therapeutics based on the interim data?
Yeah, so right now we're completing that trial. We look forward to accruing more data and releasing that to the market when we have that. The hard work that's going on right now is with the double-blind trial. So that trial is completed enrollment, but now we have the last several months of the patients completing treatment. We're going through the rigorous and somewhat arduous process of cleaning the data, ensuring its accuracy and integrity, and then we will be aiming to lock the database before we complete the formal analysis. So those are the key steps that we'll be doing between now and January when we release the data from that study.
David, thank you so much for presenting today. Have a great weekend. We look forward to hearing from you in January.
Great. Thank you so much. It's an exciting time and we appreciate all your interest.
No worries. Thank you.